Dear students, we have already had around 10 to 12 lectures in the series of lectures involving Introduction to Cell. Today we are going to start the cell theory and exceptions to cell theory and the differences between prokaryotes and eukaryotes. The cell is the fundamental structural and functional unit of all the living organism is known to all of you, and the discovery of cell was made possible with the invention of certain microscopes. Francis and Jensen in 1519 invented the first compound microscope which could magnify the substance placed underneath it 300 times. Anton van Leeuwenhoek also invented a microscope which could magnify around 270 times. With the invention of microscopes, Robert Hooke could able to see the finer things underneath the microscope, and in 1665 he discovered the cell, and what he had seen under the microscope were the dead cork cells, and he termed them cells, and thus since those were the dead cells, they did not have cytoplasm nucleus in them, and he was the discoverer of cell, and also he coined the term cell. And afterwards in 1839, Sladen and Swan they put forth cell theory. According to cell theory, the basic tenets of the cell theory are: the cell is the smallest structural unit of life, and it is also the physiological unit of all the living organisms. That means all the activities which happens in the living organisms are the outcome of the activities of its constituent cells. Also, the cells are the hereditary units. But there was a deficit in cell theory proposed by Sladen and Swan, that they could not account that from where do the new cell arises. This was meted out by Rudolf Virchow, and he said hominis celluli acellula, which means that the cell arises from pre-existing cell. The protoplasmic theory involves the development of various aspects of protoplasm. That it was first of all named as sarcode by Dujardin, and finally protoplasm term was coined by Purkinje. Von Moll said that protoplasm is the fundamental substance of all the living organisms. Max Schult proposed protoplasmic theory, that the cell is an accumulation of living substance or protoplasm limited by an outer membrane and possesses a nucleus. Inside of the nucleus, the protoplasm is termed as nucleoplasm, whereas the protoplasm which is laid outside the nucleus is termed as cytoplasm. Huxley and Hertwig stated that the protoplasm is the physical basis of life. Then the protoplasm is the complex colloidal system with living and non-living substance contained therein. The living substance is divided into nucleoplasm and cytoplasm, and the part of protoplasm which is inside the nucleus is nucleoplasm, and outside the nucleus is cytoplasm. The cytoplasm exists in two physical states. One is plasma gel and another is sol state. The sol state generally exists in the middle part, and it is called as medulla or the endoplasm, whereas the gel state exists at the outskirts of the plasma membrane, outskirts of the cytoplasm near the plasma membrane, and it is termed as ectoplasm. But both the states, they are interconvertible. Inside the cytoplasm, many organelles, which are the organs of the cell, they do exist. For instance, endoplasmic reticulum, the ribosomes, the mitochondria, the Golgi complexes, the photosynthetic units, quantasomes, which are contained in chloroplast, the cilia and flagella, which are extracellular organelles, the centrioles, lysosomes, and vacuoles are also present. However, the vacuoles are present only in plants and are absenting in animal cells except protozoans. Nucleoplasm is the part of the protoplasm which is present inside the nucleus. It contains nuclear envelope, chromatin, and nucleoli. Nucleoli, as you know, are the seat of RNA production. Thus, protoplasm contains non-living substance also, which is called as deutoplasm. All the non-living substances are collectively called as deutoplasm, such as lipid droplets, the pigments, granules, and minerals, and other substances. Come to the sizes of the cells. The sizes, shapes, they may vary greatly from kingdom to kingdom. Bacteria having a dimension of 0.2 to 5 microns, whereas ostrich egg is 6 inches in length, and nerve cell hooping 1 meter in length. Therefore, the outcome is that the cells have various sizes, and depending upon their functions, their shapes may vary also. The sizes are governed by nucleocytoplasmic ratio, the cell surface to volume ratio, and also the metabolic rate of the cell. Then, what is the significance of this cell theory? From cell theory, we could deduce that there is a synonymity and a strong relationship among the diverse living forms in the structural and functional function of the cell. Right from bacteria to man, we see some synergy in function, shape, sizes. The cells show unity in structural plan and function as well. It implies that all the living things have originated from same primitive ancestral type which arose 2 billion years ago. Now, all the cells which exist, they do not follow the cell theory. There are some exceptions to cell theory also. Like for instance, viruses and blue-green algae. Blue-green algae do not have nuclear membrane, so their nuclear material is laid, and there is no distinction between cytoplasm and nucleoplasm. Viruses, especially, they are thoroughly incomplete cells, and they are the simplest kind of cells, or in fact we cannot call them cell because they have no cytoplasm, nucleoplasm. They have simple nucleic acid in form of DNA or RNA, never both of them, enclosed in a protein coat, and they lay dormant unless they come in contact with the living machinery. They cannot reproduce or multiply, but whenever they gain access to host machinery, they complete their metabolic, they utilizes the metabolic machinery of the host cell. Like for instance, bacteriophages, which are the viruses of which infects bacteria. The cyanophages are the viruses which infect blue-green algae, and the phytophages, which are the viruses which infects or utilizes the plant cell machinery for their living and perpetuation of their species. Viruses have both kinds of characters, as I've told you, and they are designated as the connecting link between living and non-living world. So they may be having some characters which are non-living and having some characters which place them in the category of living animals. organisms. Among the non-living characteristics of viruses are that they can be crystallized like organic and inorganic compounds, but in the absence of enzyme systems, they do not show any multiplication or nor do they grow. They do not respond to the external stimuli also. These are the characters which indicates that the viruses, they are the living and non-living organisms. However, they have certain living characters that whenever they come in contact with the host machinery, specifically, they tend to multiply, and their nuclear material or their genome, they tend to undergo mutation, shows that they are living, and this is the living property. Their genetic material has the capacity to recombine, and that recombination is heritable. That shows that the viruses are living also. Now you can see this is the living cell. We'll come to it afterwards. See that viruses are the obligate parasites, means they cannot multiply in absence of host machinery. They were discovered by Ivanovsky, and they do not follow the cell theory, and the basic tenets of the cell theory were jumped by the viruses. Viruses have specific genetic program which permits reproduction in a host only utilizing its machinery. Their genetic material undergoes heredity and mutations. They lack cell membrane, metabolic machinery, and biosynthetic machinery, but they may have certain enzymes which functions whenever the virus come in contact with the host machinery. Tobacco mosaic virus was the first virus which was discovered by Ivanovsky, and this virus was crystallized by Stanley, and he said by this property displayed by the virus, Stanley stated that viruses are the connecting links between living and the non-living worlds. Viruses are obligatory parasites because they totally depend upon the host cell. Outside the host cells, they are inert and inactive. They can be crystallized, as I have told you, and can be stored in bottles like crystals. They utilize biosynthetic machinery of host for the synthesis of their own nucleic acids and proteins. However, they are so small that they can pass through porcelain filters through which bacteria could not pass. If we see the general structure of the virus, they have a dimension of around 30 nanometer to 300 nanometers. They consist of this central core of nucleic acid, which is surrounded by a protein coat which is called as capsid. The intact virus which has the capacity to infect is called as virion. This capsid, this virion is the assembly which contains nucleic acid as a core material and capsid as a surrounding material. This capsid is made up of protein, and its subunits are called as capsomeres. Capsid is the outer coat of protein made up of subunits called as capsomeres. These are arranged helically or in form of polyhedron in animals. In many animals, animal viruses like influenza, herpes, mumps, smallpox virus, and few plant viruses, they have lipoprotein coat also around them. Capsid protects the host genome from too much of mutation, and the nuclease enzyme, because if protein coat is absenting, the nuclease enzyme could digest the nuclear material of the virus and they would die. This capsid has certain properties that it enables the virus to bind to the cell surface. It has another property that it acts as enzymes to dissolve the surface layer of the host cell for penetration of the virus or penetration of the nucleic acid inside the host cell. Viruses have different kinds of nucleic acid. DNA virus have deoxyribose nucleic acid, which may be double-stranded or single-stranded. The double-stranded can be linear or circular. Like T5 and T7 viruses, they have linear double-stranded DNA, and circular double-stranded DNA is found in polyoma and SV40. Single-stranded DNA viruses are also present, linear parvovirus and circular SciX174. RNA virus again are double-stranded like reovirus, and single-stranded like tobacco mosaic and influenza viruses. All the plant viruses and a few animal viruses like polio, mumps, measles, and cold and influenza virus, they have RNA as genetic material. This is the diagram of bacteriophage. I have already told you what are bacteriophages, the viruses which infects the bacteria. There are viruses which infects animal cells and plant cells as well. And bacteriophages which are parasitizing the bacteria, are discovered by D. Harrell in 1970, and they have a look or hangover like a tadpole larva. You can see here that the head and the tail, two parts are there. Head contains capsid, and inside the capsid is a nucleic acid which has very scanty number of genes, around 75 genes only present, and it is only 53 microns long. Only 2,000 capsomeres are present in the capsid. Tails have tail fibers which are polypeptide chains or proteins. The tail can be contracted and elongated, and this part is attached to the cell wall of the bacteria, and the hollow part is inserted inside the cell wall, and the viral material is injected. The material means the nucleic acid is injected in the bacteria. So this is how bacteria they infect this is how the viruses they infect the bacteria. The head part is hexagonal, as you can sight in the plate. Its capsid is made up of around 2,000 capsomeres, each of around 80,000 Daltons. The tail is in form of hollow cylinder. Around 144 subunits of proteins are there in it. Rests on an end plate, and tail fibers are bundle of polypeptide chains, probably meant for anchorage on the cell wall of the bacteria. The animal viruses are reoviruses, oncoviruses, smallpox virus, chickenpox, rabies, polio, mumps, measles, influenza, hepatitis, and cold virus. Whereas plant viruses are tobacco mosaic virus, the TRV virus, tobacco rattle virus, tobacco mosaic virus, potato virus, etc. So, students, tobacco mosaic virus was first discovered by Ivanovsky and was isolated in crystalline form by Stanley, as I've told you in this lecture. Now, another more simpler infectious agents are also present, which are called as viroids. They are simpler than viruses and are made up of single RNA molecules only, and the protein coat is absenting, produces disease in plants. So they are even simpler than viruses, still they could infect. So this is the cell theory and the exception to the cell theory. Now let us see how the cell it looks. you can view in the slide the ultrastructure of the animal cell. This the outer limiting membrane is plasma membrane. It is thrown into folds which are called as microvilli. This probably increases the this actually increases the surface area of the plasma membrane, meant for increases its absorptive capacity. And from the blepharoplast, cilium arises, and you can see the nucleus, and which is bound by nuclear membrane. Chromatin is present inside the nuclear membrane. In the nucleoplasm, chromatin is nothing but DNA in association with the histone proteins, or polyamines in prokaryotes, whatever the case may be. However, prokaryotes, they do not have cell membrane around them. After the nuclear membrane, we have endoplasmic reticulum, which on which you can view some ribosomes. These are called as rough endoplasmic reticulum. They actually are the seat of protein synthesis and processing of the proteins produced therein. After the endoplasmic reticulum, we have Golgi bodies, which are meant for secretion and processing of the protein produced. The ribosomes in the eukaryotes, they are of 80 Swedberg unit type, whereas in prokaryotes we have 70S type of ribosomes. We have mitochondria as the powerhouses, which provides energy and are the seat of production of ATPs, which are the energy providing molecules. We have desmosomes, which are meant for anchorage of two neighboring cells. And this is the basic general structure of eukaryotic cells. So dear students, this is all in this lecture. Thank you so much.